Hello dear students. So today we are moving on with the same chapter, that is sexual reproduction in flowering plants. So in the previous session we had studied about pollination, different types of pollination, and also different agents of pollination, the agents which help in pollination. So all that in detail we studied. Okay. So today we are moving on. Okay, we have to study about outbreeding devices. Okay, so what is this? Now in the last session and all, we studied that flowering plants, they produce bisexual flower. So majority of the plants will be producing bisexual flower. So you know the meaning of bisexual flower, that the flower has both androecium and gynoecium. So when that is the case, there is more possibility of self-pollination. Means a flower has anther and stigma close by, and when pollen grains are released, there is more chance that within the same flower the pollen grains may fall on the stigma and bring about pollination, that is fertilization may happen. Okay, so that is called like self-pollination. So continuous self-pollination, if it happens, it leads to inbreeding depression. So about that in detail you will be studying in the coming chapters. Okay, so continuously if there is self-pollination, there will be like no variety, and also inbreeding depression, okay, is like continuous self-pollination. What happens is the plant becomes, the varieties will be less, okay, and they'll be like more prone to diseases, the resistance, like all that will be affected, and we call it as inbreeding depression. Okay, so to avoid that, we see that plants will actually have some techniques to avoid self-pollination. So even though the plant is producing a bisexual flower, okay, it will avoid self-pollination. So what are those techniques, okay, developed by the plant to prevent autogammy? So you know we use the word autogammy for self-pollination. So to avoid autogammy, flowering plants develop certain techniques, okay, to avoid autogammy, and that is called as outbreeding devices to prevent autogammy. So we will see one by one what are the different techniques developed by the plants. Okay, so the first one is unisexuality. So this unisexuality means the plants will produce only one type of flower, either female flower or only male flower. So one plant is producing only male flowers, another plant is producing only female flowers, then self-pollination or autogamy can be reduced. Okay, that is unisexuality, or we can also call it as diclaning. Okay, so this unisexuality we see like example papaya plant. Okay, so male plant is separate, female plant is separate. So by this autogamy can be prevented. So next we see dichogamy. Okay, so dichogamy is nothing but the maturation of androecium and gynoecium is not happening at the same time. There is no synchrony between the maturation of androecium and gynoecium. So about this I had told you in the previous session also. This is one of the technique where the plant can prevent autogamy. Even though it's a bisexual flower, androecium and gynoecium is present in the flower, okay, but androecium is maturing at a different time, gynoecium is maturing at the different time. So by this the plant can avoid autogamy. Okay, then we see heterostyly. So heterostyly is a technique where anther and stigma are placed at different positions. So within the flower, okay, the position will differ. They are not in the same plane. They are not in the same level. At different position it is present. So by this okay, to some extent at least autogamy can be prevented. Okay, that is heterostyly. Then coming to self-incompatibility. So self-incompatibility is a genetic mechanism, okay, where the plant, the stigma will recognize the pollen of the same plant. Okay, it can recognize that and it will not allow the pollens to germinate. Okay, so we say this is a genetic mechanism. So even though plants are having bisexual flower, okay, pollens fall on the stigma, but it will not be allowed to germinate, and that is called as self-incompatibility. Okay, so I told you it's a genetic mechanism. And the last one is hercogamy. So this hercogamy is there is like some barriers, okay, so some physical barrier is present in between androecium and gynoecium, that is between the stamens and the carpels. Some physical barrier will be there, okay, by which autogamy can be prevented. Okay, so these are some of the techniques which are developed by the plant, okay, to prevent autogamy. This is a probable 5 marks question. Okay, so then you need to write all these 5 points with a sentence of explanation. Okay, so I told you this is a different techniques practiced by the plant to prevent autogamy. Okay, now after that we are moving on to the next part of the chapter, that is pollen pistil interaction. Okay, so now we have studied about pollination. Pollens are released from the anther, okay, and it is transferred to stigma. So that is pollination. Now after that what happens is the pollen pistil interaction. Now just we say pollination. Now pollination will not ensure or it cannot guarantee, okay, the right type of pollen to fall on the stigma. Now what do we mean by right type of pollen? That is the pollens which are compatible to the plant. Okay, many a times pollen grains of the wrong type may fall on the stigma. Now what do we mean by this wrong type of pollen? So either it may be pollens of different species or it may be of the same species but incompatible ones. Okay, so these pollens are called as incompatible ones, and if this, that is it may be a right pollen or a wrong type of pollen falling on the stigma. But the stigma has the ability to recognize the pollen whether it is of the right type or if it is of the wrong type. Okay, so if the pollen is of the right type, then the stigma, okay, so you can see a diagram here. I've just shown the topmost part of the stigma. Okay, I have not continued with the style here. This is only stigma with pollens which are deposited, landed on the stigma. So if the pollen grains are of the right type, okay, then the stigma will accept the pollen, okay, and it will be continued with all the post pollination events. But if the pollen grains are of the wrong type, I told you what are the different types of, like when we say wrong pollens, what are they? So if the pollens are of the wrong type, then the pollens will be rejected. Now what do you mean by rejection? Okay, so rejecting means these pollens of the wrong type will not be allowed to germinate. Okay, so when we say it is not allowed to germinate, it means it will not be allowed to produce the pollen tube. Now you can see here these are all germinated. Okay, so if it is of the wrong type, it will not be allowed to germinate. It will not be allowed to produce the pollen tube, or it may just begin to produce the pollen tube, but the growth of the pollen tube will be stopped. Okay, so this is how what happens, that is if when stigma can recognize the right type of the pollen and the wrong type of pollen. Now how does this recognition take place? How does it happen? So we say the stigma can recognize the right type of pollen by, as a result, okay, as a result of there is a communication or discussion between the pollen and the stigma. Now when we say communication, what type of communication or what type of interaction? It is the interaction between the chemicals of pollen grain and the chemicals of the stigma. Okay, so the chemicals of the pollen grain, that is the pollen grain surface, and the chemical components of the stigma, there is an interaction. By this interaction, the stigma recognizes the right type of pollen and the wrong type of pollen. So there's a, you can say there's a chemical communication between the pollen grain and the stigma. Okay, so it was just in the recent years that the botanists have done a lot of work over this, and they got a lot of information about the different components of pollen grain and different components of stigma. So how that interaction goes about and all that, we could get the information. So once the right type of pollen grains fall on the stigma, I told you the pollen grains will germinate. So when we say germinate, what exactly happens? Let's see. Now this is the structure of a typical pollen grain. So I have taught you this in the last previous sessions. Okay, so it has an outer exine, next to that another layer of intine, okay, and a vegetative cell and a generative cell. So when the pollen grains, when the right type of pollen grains fall on the stigma, okay, it will begin to germinate. That is, you will see that through one of the germ pores. Okay, so exine has pores or apertures which we call as germ pores, or in simple words we can say openings, which are called as germ pores. So through the germ pores, you will see pollen tube will start arising. Okay, so pollen tubes will start arising, and you will see that all the contents of the pollen tube also will start flowing into the pollen tube. Okay, so now you can see here that is a stigma. Many pollens are there, and some pollens have already begun to germinate. So when they germinate, this is what is happening. That is, the pollen grain, okay, through the germ pores, you will see that the pollen tube will start arising. So as it arises, I told you now, this whole content of the pollen grain will start flowing into the pollen tube. Okay, now you have to just recall another concept here. When I explained you about pollen grain, okay, structure, I had told that 60% of angiosperms are shedding the pollen grains in 2-cell stage, and remaining 40% of the plants are shedding the pollen grains at 3-cell stage. Okay, so you have to just recall that to understand what happens next. Now when it is 2-cell stage, it is like this: one vegetative cell, one generative cell. Okay, when it is a 3-celled pollen grain, it is like this. It has one vegetative cell and 2 generative cell. Okay, so these two are the generative cell, whereas here you can see there is only one generative cell. Okay, and you should remember that the nucleus of the generative cell are actually the male gametes. Okay, so the nucleus of the generative cell is the male gamete. Okay, so confusion arises here. This pollen grain has only one male gamete. In this 3-celled pollen grain, there are 2 male gametes. So how come? How can this happen? So here I want to say one thing. Now if it is 2 pollen grain or 3-celled pollen grain, okay, finally every pollen grain will have 2 male gametes. So how does that happen? Now we'll just take an example of 2-celled pollen grain. Okay, so here there's only one generative cell. So when this type of pollen grain germinates, Okay, I told you all the contents will be flowing into the pollen tube. Okay, and as this grows, as the pollen tube grows, you will see that within the pollen tube, this generative nucleus will divide into two. Okay, so this division is happening in the pollen tube. As the pollen tube grows through the style, you will see that the division of the generative nucleus take place. Now what happens if this pollen grain is germinating? Now this pollen grain, if this type, that is if 3-celled pollen grain is falling on the stigma, all the contents are flowing into the pollen tube. Okay, there is no division taking place because already it's done. Okay, already the division is over and you will see 2 generative cell already present there. Whereas in this type of pollen, this division is happening late. That's the only difference, but finally, okay, it is 2 male gametes, that is 2 generative cell will be present. Okay, so here the division is happening later, whereas in this type of pollen grain, that division is already done. That's the only difference. Okay, so if it is 2-celled pollen grain, generative cell is dividing into 2. So generative cell means the generative nucleus. Okay, you should keep in mind that they are nothing but the male gametes. So all these contents flowing into the pollen tube, okay, and generative nucleus is dividing into 2. So when the right type of pollen grains fall on the stigma, they will begin to germinate, or they will begin to produce pollen tubes. Now in the first diagram here, this is LS of the ovary or the LS of the pistil, which is showing the detail of the pollen grains and the pollen tubes. So here you will see this is the stigma. On the stigma you can see many pollen grains which are landed. Few of them are germinating, few are not. Okay, and you will see here the pollen tube. Now this is the pollen tube. Okay, so one pollen grain will be finally the pollen tube reaching the ovary. So you can see this long structure which is a pollen tube. So how the pollen tubes are produced, I have already explained now. So you know that the pollen tube is carrying all the contents of the pollen grain. Okay, so the cytoplasm, the nucleus of vegetative cell, nucleus of generative cell, so all that is moving into the pollen tube. So this pollen tube is moving through the style. It will dissolve the cells there, make its way through the style, and finally the pollen tube will reach the ovary. Now in the ovary you know that there is a presence of ovule. So this is an anatropous ovule which is like bent downwards here. So ovule has an opening called as micropyle. So here you will see the opening called as micropyle. So this pollen tube grows here and reaches the micropyle. Okay, so when it reaches the micropyle, so meanwhile this one, the pollen grains which are of 2-celled stage also will divide, that is the generative nucleus will divide and it will have 2 male gametes. Okay, so the pollen tube is reaching the micropyle. So when it reaches the micropyle, now here in the second diagram you can see this is only the egg apparatus which is enlarged and shown. We are not showing the whole ovule here, which is concentrating on the egg apparatus. So the female gametophyte, only the lower half has been shown here. So this is the pollen tube which is reaching the female gametophyte. So once it enters this opening called as micropyle, okay, so through this it will reach the female gametophyte. So when it is reaching the female gametophyte, you can see here there are 2 main nuclei, the important nuclei, which is male gametes. Okay, so this is nothing but it's the nucleus of which cell? It is the nucleus of generative cell. You also find the presence of vegetative nucleus also, but they are not taking part in the fertilization. these two, that is the male gametes, are taking part in fertilization. So this pollen tube, which is carrying these 3 nuclei along with the cytoplasm, it is reaching the micropyle, okay, entering into the female gametophyte. See here you can see it is just entering into the female gametophyte. Now there is one more thing you need to remember here. Egg apparatus has a set of 3 cells. One is egg cell and 2 other cells called as synergids. Okay. Now in the synergids, these finger-like projections, what you find here, it is called as filiform apparatus. Okay. So this structure here, we call it as filiform apparatus. Okay, so this filiform apparatus is present in synergids. Now if you observe here, the pollen tube is actually reaching the filiform apparatus. So I had actually told you in the previous session also that the main function of filiform apparatus is to guide the pollen tube into the female gametophyte. Okay, so always the pollen tube will make its entry into the female gametophyte through the synergid cell, specifically through the filiform apparatus. So that's a 1 mark question. What is the function of filiform apparatus? So first you need to remember, where is filiform apparatus? It is in the synergid cell. Okay, and the importance of filiform apparatus is it will guide the pollen tube into the embryo sac during fertilization. Okay, so the whole process from the time, that is after pollination, the pollen grains fall on the stigma. The stigma will recognize the right type of pollen. It will allow those pollens to germinate, and the pollen tube arises, which reaches the ovary, reaches the micropylar opening, and also it will make its way into the embryo sac. Okay, so once it enters the embryo sac, it will release the male gametes. Okay, so till here we say it has pollen pistil interaction. Okay, so what happens after this is fertilization. Once the male gametes are released and once they fuse, it is the next main event, that is fertilization. So from the time pollen grains fall on the stigma till the male gametes are just discharged into the female gametophyte, this whole event we call it as pollen pistil interaction. Okay, so here you will see 2 male gametes. Okay, so 2 male gametes are nothing but the nuclei of generative cell. Okay, so they are, so in the third diagram you can see the release of male gametes. Okay, so 1 male gamete will be entering or moving towards the egg cell, and 1 more will be moving towards the central cell. Okay, so till all these events, the whole thing, we call it as pollen pistil interaction. Now this germination of pollen grain, very easily we can see by using some common flowers like vinca, okay, or crotalaria. We can take the flowers, collect the pollen of these flowers, we can take a slide. We need a medium in which we can artificially make the pollen grains to produce the pollen tube. So what we do is we take sucrose solution, 10% of sucrose solution we take on a slide. Okay, we can dust some pollens onto this sucrose solution. We can leave it for around 20 to 30 minutes. And after that, when we observe under the microscope, you can see that the pollen grain would have germinated. That is the production of pollen tube we can see. So that's a very simple experiment which we will be doing in our practicals also, where we will make this pollen grain to germinate and we can observe the pollen tube very clearly. So after getting the whole information about pollen pistil interaction, so about the different chemical components and the whole information, it has been used in plant breeding. Okay, so about that in detail you will be studying in the further chapters. Okay, so a plant breeder, he will be trying to cross different plants of different species or even different genera also. Okay, so the aim is to get a hybrid variety. So there what is the main concept is to bring all the desirable characters in one plant by crossing different plants. Okay, so the desirable character has to be got in one plant which can be considered as like a superior variety, a good variety which has all the desirable characters. So that can be achieved by having the information about all this. Okay, so we are going to now study about like how it is used for crop improvement programs. Okay, that is artificial hybridization. Okay, so artificial hybridization, it is by knowing the information about which pollens has to be used. Okay, by using which pollen we can actually cross two different plants to get a desired variety. Okay, so all that is followed under this. So what is done here is a female plant. Okay, so a female plant may also produce a bisexual flower only, but it is considered as a female plant. Okay, so when we consider a plant as a female plant, okay, from those flowers of that plant, we have to just remove off the androecium. Okay, because pollen grains of another plant will be selected, so that plant will be selected as male plant. Okay, so a female plant, even though it produces a bisexual flower, what is done is, as the plant grows, when the flower is still in a bud condition, what has to be done is the anthers has to be removed. So anthers have to be removed before it matures, that is before it produces the pollen grains, the anthers will be removed, and this step we call it as emasculation. Okay, so in artificial hybridization we select a female plant, okay, the female plant bearing a bisexual flower, and in that flower the anthers should be removed before maturation, and this step we call it as emasculation. So once emasculation is done, then this flower has to be covered. So it has to be covered for what reason? Because pollens of some unwanted plants also may come and fall on the stigma, which we don't want, because we want our desired selected pollens to be dusted over that. Okay, so because of that, what is done is, after emasculation, the flower is covered by a bag usually made up of like butter paper. Okay, it is covered, and this step is called as bagging. Okay, so once it is bagged, it is just allowed to grow. So after it reaches maturation, once the flower is matured, then what is done is the pollens of the desired plant is selected, it is brought and it is dusted over this plant. The flower which is being bagged, the bag is removed and the pollens of the desired plant is dusted over that. After dusting, again it is covered so that we don't want any contamination of any other pollen grains. Okay, again it is bagged and it is just left to grow. So what happens is, the pollen, that is the desired pollens which are dusted over the stigma, it will germinate, produce the pollen tube, fertilization happens, and finally fruits and seeds are produced. Okay, so now this is the case if it's a bisexual plant, that is if the plant is producing a bisexual flower. If it is a unisexual plant, male plant is separate, female plant is separate, so this step is not required. Emasculation is not required because the flower already has only one reproductive structure, either male or female. Okay, so the female plant is selected, no emasculation required because the flower will have only the female part, that is the carpel. But this has to be done. bagging will be done because we don't want any contamination of undesired pollen, and the remaining process remains the same. So with this, there is a possibility of getting a hybrid plant. Okay, so I told in the beginning, hybrid plant or hybrid variety where we want to combine all the desired characters to get a superior variety of plant. Okay, so with this information, as I told, it is in the crop improvement programs, it has helped a lot, and this knowledge has been used practically. Okay, so that is about artificial hybridization. Now this part, artificial hybridization, can be asked as a short note. Okay, or a short note where you need to just brief out all these steps. Okay, so that is about artificial hybridization. Okay, so now we have studied about pollen pistil interaction. Okay, so pollen pistil interaction, that is till the gametes reach the female gametophyte. What happens next is the main event which we call as fertilization. Okay, so this fertilization is a main event which we will be studying in the next session. Okay, so in today's session what we studied, okay, so all this, that is pollen pistil interaction, the outbreeding devices, as I told you, it's a probable 5-mark question. Okay, so I want you all to practice that 5 points. I want you to practice. Okay, and I want you to write the answer, and as I say in all the videos, okay, there's a link below the video where you can take a pic of that and post it. Okay, so we'll continue the next session. Thank you. Have a good day.